Professor Adrian Liston, Professor of Pathology here at the University of Cambridge, and today I'm going to be talking about diversity in the immune system. I want to start by just saying, what is an immune system? And essentially, an immune system is anything that can be used to prevent infection by microbes. And all organisms have immune systems. Even single-celled bacteria actually have sophisticated immune systems. You might have heard about a technology called CRISPR, which is used to genetically engineer new therapies. That was actually evolved as an immune system for bacteria to stop them being infected by bacteriophages, a type of virus. Now, humans have much more complex immune systems than bacteria. We actually have around about 100 different types of immune cells, and there are 1.8 trillion immune cells in our body, with 10 billion new immune cells being made every day. This includes a lot of cells in our blood, our lymph nodes, our bone marrow and spleen, but also low numbers of immune cells scattered through essentially every organ in our body. And the immune system is comprised by about 2,000 genes, or nearly 10% of the human genome being involved in immunity against infections. The immune system is also extremely energetically expensive. It takes a lot of resources for us to run this immune system. All of those cells I spoke about a second ago, together, they only weigh about 1.2 kilograms. It's not a huge part of your body. But that 1.2 kilograms of immune cells, they use around 380 calories every day, even when you're completely healthy, which is about 18% of your energy consumption. And if you're fighting an infection, they can be using 1,400 calories a day, which is about 69% of your daily energy consumption. That's essentially your immune system using 5 Big Macs worth of calories every day that it's fighting an infection. Now, the immune system is not simply an on-off switch, and you'll often see drinks like this or foods that claim to be immune boosting. And even after studying the immune system for decades, I'm still actually not sure what it is that they're claiming. Claims like immune boost, they imply that the immune system is a simple dial that can be amped up. And this is simply not how the immune system works. We don't have one immune system that gets boosted up. We have hundreds of immune types, and that means that we can have many different types of immune reaction that can occur. So when someone calls a product immune boosting, which cells are they actually claiming are boosted? Because there are different immune responses that can occur. In fact, I think it's much better not to think of the immune system as a single system. There is no central decision. There is no central outcome. Instead, I prefer to think of the immune system as 1.8 trillion battle robots, each individually processing information and each individually deciding how to respond. Now, these cells are all making their own decisions, and they can be working with each other or against each other. But the net effect of all of these cells making their own decision on how to respond does come to some type of organic decision making. And the cells tend to coordinate around a loose consensus of different types of inflammation. We have type 1 inflammation, type 2 inflammation, type 3 inflammation, and just when you think you know what's coming next, we have humoral immunity. We also have another type of immune response that is much more orientated around repair and regeneration. But really the key point here is that inflammation is not inflammation. There are many different types of immune response that can occur that involve different cells and that have different impacts on any infectious organism. Now, today's topic is diversity in the immune system. And I think it's really important to note that all biological systems are variable. Even simple traits like height have a bell curve. You have people that are around about average, some people who are extremely short, some people who are extremely tall. Now, the immune system, though, this is really hypervariable. It's more than 10,000 times more variable than other traits you might think about. And if the immune system was height, the amount of variation that we have from person to person would be equivalent to the tallest people being able to step over Mount Everest, while the shortest people would be shorter than the width of a human hair. That is massive amounts of variation. So why is the human immune system so diverse? The answer, as is so often the case in biology, lies in evolution. And evolution is essentially a competition. Now we can think about a predator-prey relationship, such as the cheetah and the gazelle. And here we'll have a race between predators and prey. The cheetah wants to run fast enough so that it can catch the gazelle, and the gazelle wants to run fast enough that it can get away from the cheetah. Now, if we think about the gazelle, you're going to have a bell curve. You're going to have some gazelle that are a bit faster and some that are a bit slower. The slower ones are going to be eaten by the cheetah, which means on time, the population is going to get faster. At the same time, you've got a bell curve of cheetahs. The faster cheetahs are going to catch the gazelle. The slower cheetahs are going to go hungry. So over time, the speed of the cheetahs get faster. And again, as the cheetahs get faster, so will the gazelle get faster. And in fact, these two are locked in what's called an evolutionary arms race, where both populations are getting slightly faster each generation. Of course, there's not just one way to avoid predation. There can be speed, there can be strength, there can be intelligence, and you can be optimizing different strategies depending on what predators are mostly trying to get you. Now, once we start thinking about infections in humans, this is essentially a predator-prey relationship where we are the prey and the microbes are the predators. The problem is that this is a competition that humans simply can't win if we play by the rules. Every generation, both sides are going to get stronger, but for humans, a generation is 20 years. Whereas for a bacteria, for example, a generation is 20 minutes. They can just go through so many more generations than we can. Their evolution is in turbo speed compared to our evolution. So the solution has to be something different. If we simply act like gazelles and we try to go faster than the bacteria, which is acting like a cheetah, every generation, those bacteria are going to get better and better and better at beating our immune system. And very rapidly, they're going to win consistently. Instead, our solution that has been forged by evolution is for different individuals in the population to take different strategies. So just as you've got gazelles and meerkats and wildebeest with very different strategies about evading predators, we will have some individual humans might be like a gazelle. Others, though, will be like a wildebeest or a meerkat. And so if a bacteria starts evolving and is trying to get around a particular strategy, say the gazelle strategy, it'll get better and better and better until it hits an individual that actually has a wildebeest strategy. And then their bacterial strategy doesn't work anymore. You might go to another individual who's got a meerkat strategy, and again, the new evolved strategy doesn't work anymore. So the more variable we are from person to person, the more we stop our microbes becoming hyper-specialized about beating our immune response. So really, diversity saves the human population from infections. The more diverse our immune systems are, the less pathogens are able to specialize and the more that we can beat them. Now for the next part of the talk, I want to bring up a concept that I call immunological space. Now we've got these different types of coordinated immune responses, and at its absolute simplest level, we can consider immune responses of 4 different types, the type 1, type 2, type 3, the humoral immunity, and we can just consider a map where different places on that map are people who are better or worse at the different types of immune response. So here you've got 2 individuals, a red individual and a blue individual, And one of them is a bit better at type 1 and type 2 immunity, and one of them is a bit better at humoral immunity and type 3 immunity. So what I want you to imagine is that individuals that are moving around on this map are actually moving into slightly different immune constellations, being better at one type of immune system in one part of the map, and if they move to another part of the map, that's trying to represent that they're getting better at a different type of immune response. Now, what we know about the diversity in the immune system is that it's pretty stable. The individuals will shuffle around a little bit from day to day, even week to week, but overall they tend to stay in around about that same spot. If someone is pretty good at type 2 immunity, they tend to be pretty good at type 2 immunity 6 months later, a year later. And the reason we know this is we've gone and tested it. We've taken 200 plus individuals, we've measured their immune system every 6 months, and then go back and say, how similar is their immune system over time? And we find that while there's a lot of variation between individuals, there's very little variation over time within a single individual. We also know that the immune system profile is quite resilient. Even if you have an infection and it radically changes your immune system, after that infection, people tend to fall back in their original profile. So you might be very good at type 1 immunity, and if you have an infection, you might nip off and have a strong type 3 immune response, but then afterwards, you'll go back to your type 1 baseline setting. Now, we found this out through a study that we'll call the Fun Holiday Study. Here we took 50 individuals that were planning to travel to a high-risk country for gastroenteritis. We took a sample of their immune system before they went on holiday. Then we said, go, have fun, try the street food, and come back when you get home. They went away, and about half of them got acute gastroenteritis while they were on holiday, and half of them were continuously healthy. And we saw how much did their immune system change during that holiday. And the answer is not much. And there wasn't any difference between people who were continually healthy or people who had this big immune challenge and then came back afterwards. Now, the final part of the talk is about what are the drivers of the diversity in the immune system. We've said that the diversity of the immune system is important. We've shown that it's stable and resilient. But what actually makes one person's immune system different from another person's immune system? Well, here there are knowns and unknowns and known unknowns. We know that genetics is important. In fact, about 30% of the variation is probably caused by genetic differences. And here we can really take advantage of work from identical twins, where we can separate identical twins that have got the same genetics and fraternal twins that have got similar genetics, but not to the same degree as identical twins. And if we do the calculations on how much immune variation we see, about 30% of the variation boils down to genetics. Now, for very few of these do we actually know which genes, but we do know that it is genetic in basis. Age is also quite important. About 10% of the variation from individual to individual can be explained by age, and this age change is pretty predictable over the course of decades. And then we also have the effect of environment. And environment is really quite powerful. It's about 50% of the variation that we see. And this comes from a study that we did that we call the parenting study. So here we took 138 cohabiting individuals. So we were able to take parents of a young child at home, and we compared the immune system of the two parents. And here what we did is we profiled how different the parents were from each other. And what we can see on the left are people in immunological space, and you can look at the mother and the father in each pair, and you can see how close they are. And there are some people that are really outliers from most of the population, but they're relatively close to each other. And you can work out the average distance in terms of the immune system between the parents. Or what you can do is take those parents, split them up, and then pair them up with random adults. Now, of course, We don't actually ask them to get a divorce and get remarried and study their immune system, although that would actually be a fantastic study. Instead, what we do is we just do an in silico randomization. We just randomize and see how different they would be if we paired them up in a computer with someone else. And what we see is about half of the variation that you would have between random individuals disappears when you look at parental pairs, which means that living together is eradicating about half of the difference in the immune profiles from individual to individual. So we can imagine this as two individuals, their immune system is slowly changing over decades, and now, oh look, they're going to meet, fall in love, their immune systems are converging as they start to share the same environment, and now they have a closer immune system than they originally did. Now the final thing I want to talk about here is sex. So sex is actually not that important a variable in terms of driving immune diversity in humans. It's responsible for about 5% of variation. Although I should say this 5% could be sex. It could be due to sex hormones, XY chromosomes, anatomical development, but it could also be gender. And here, when I'm talking about gender, I'm talking about really different environmental exposures for men and women may be driving some of the difference in the immune system that you detect when you measure the immune system in men versus women. Now, it's really actually quite difficult to dissect sex versus gender. And this is because there are confounding factors. You've got the biological sex, but then the gender is the exposure differences that are due to culture. Now, to explain this, let's consider the case of lung cancer. If we were to look at the rate of lung cancer in 1980, we would say there's a very strong sex bias. There are many more men with lung cancer than women with lung cancer. However, we now know that lung cancer is heavily driven by smoking, and it arises about 30 years after smoking. So if we look at the smoking rates in 1950, we see that there's a huge gender bias in smoking between men and women. And in fact, that more or less matches what we thought was a sex bias in 1980s lung cancer, and that we would now describe as a gender bias. This big sex difference between males and females in 1980s lung cancer rates was really due to gendered smoking rates in the 50s. Now, if we look at smoking in the 1990s, here the gender gap has really shrunk down, fairly equivalent numbers of men and women smoking in the 90s. And if we look at lung cancer rates today, in 2020s, we see that that sex bias has really shrunk down as well. So a lot of that effect probably was actually gender and not sex. And there are many, many different environmental chemicals that have got different exposures by gender or race. For example, aniline dyes in textile factories. This really drives bladder cancer in factory workers, most of whom are men. Or radium in watch dial paint. This caused radium jaw in watch painters, which are almost all women, causing the phenomenon of the radium girls, as they were called at the time. You've also got products such as toluene in hair products, which cause contact dermatitis in hairdressers. And again, the gender difference in hairdressing as a profession leads to this gender difference in dermatitis as a disease. So how much of what we see as sex is actually gendered exposures? Now, I'd love to answer this conclusively, but we don't actually have much data. One of the best studies that's come out was a systems immunology study done in Morocco. Now, there's two parts of the study. I'm only going to show you the first part at the start. And what we've got is men and women studied for their immune profile in Morocco. And you can, if you're looking at where those men and women are in terms of immunological space, you would say there's a huge difference. Clearly, two separate populations with men and women having a different immune profile. However, what I've done here is I've left off one key factor. This population of men and women were studied in the rural setting, in a very traditional rural setting that had strict segregation by gender. If we look at a second population of men and women, also in Morocco, but in an urban setting, the story changes a lot. You see that now the men and women from the urban setting have a very, very minor sex effect. And this means that most of the sex effect that we saw in that traditional rural setting wasn't actually a sex effect. It was an effect due to behavioral segregation. In other words, it was gender. So just to sum up this talk, the immune system is absolutely amazing. It's literally 1 trillion cells that can self-organize into a killing machine optimized to kill any pathogen that comes near us. And humans have a hypervariable immune system, so much more different from individual to individual than any other trait you might think of. This is a really good thing. Yes, it means that we have got a different susceptibility to immune disorders and a different susceptibility to different infections, but it stops the pathogens from hyper-specializing and killing us all. Now, our personal immune response is quite stable and quite resilient, and it's shaped by our genes, our environment, and our age. There's also a minor role for sex, although I think it's probably overestimated and gender is likely to be much more important. But really, there's a huge amount we still don't know about the immune system. With that, I'd like to thank you for your attention, and I'd really like to thank the people in my lab that are working on these problems and many other interesting immunological problems. If you've enjoyed this talk, consider donating to support our innovative community engagement projects that we're running at the moment. For example, we're just preparing a new book on becoming a scientist, where we walk through the really fascinating stories of all the individuals in our lab, the challenges they overcame to become a scientist, and the motivations they had to do so. Thank you.